Thank you so much, Lisa. This is, and Zoe, this is an absolute honor to be part of this conference. Actually, I'm just realizing, can you all hear me okay? Because I don't have my headphones in. Is this okay? All right, very good. So I will say I have a cold right now, so I'm hoping that I don't have to step off and blow my nose at any point in time and my voice stays good. But if anytime you can't hear me, do let me know. I'm happy to take questions at any time. And really the goal of this talk is just to highlight some different directions in mathematical biology. I'm specifically going to talk about some projects that students have been involved in. I'll mention that I am recruiting students for REUs for the upcoming summer. And really, we are only looking for coding experience. You don't have to have taken calculus or differential equations or linear algebra. So you can be a first-time researcher. And yeah, I'll also stick around during the networking lunch. I'll probably be in the REU room. So I'm happy to chat with folks there as well. I'm going to upload these slides to my website. I forgot to do that beforehand, but I'll put them on my website underneath resources so you'll have access to all this stuff. And so both the videos that you see on this front slide were actually created by students. So this one was developed by Annapurna, who is a current junior in my group, and this one was developed by a team of students a few years ago. Okay, so I figured since we're going to be spending the next hour together that I'd introduce myself. So outside of math, this is Mr. K. I like to joke that he is teacher's pet, but he really likes his fashionable neck ties and bows. And then outside of math, the other thing I really like to do is actually make flower arrangements. That's kind of how I find my calm and peace, and I listen to audiobooks on repeat. So I've just kind of moved around the US. I did my undergraduate degree at the University of Maryland, Baltimore County, so a liberal arts public school. And I really have gone from math to applied math. I did an applied math PhD, to math bio for postdoc positions, and now I'm in a math department. And I chose math probably halfway through undergraduate, and it was partly because a mentor of mine, Dr. John Zweck, said that medical doctors needed mathematicians because we could comment on the problems in new ways. And so that really inspired me, this idea that I could contribute to biological questions from a different perspective. And now, as I've kind of grown, the other reason I kind of stuck with math is really because it's allowed me to not decide what I want to be when I grow up, in some sense. It really lets me work on lots of different application areas, whatever I'm interested in. So I work on lots of different stuff in collaboration with experimentalists, often ranging from plant development to animal skin patterns, and also a little bit in the social sciences related to, for example, social movements on Twitter or now X, and some work on election forecasting. And so all these different areas might seem odd and not related at first, but really they all fit into this kind of umbrella of what's called complex systems. So complex systems are settings where you have individuals that come together to create some collective group dynamic that is bigger and richer than the individual pieces. So my personal favorite example of a complex system is one that I have not worked on yet. I'm going to start this video in just a moment to highlight it. But really, to explain this, this is a circular road. It's not a real road. It's an experimental setup. And what happened was that these cars were placed equidistantly on the road, equal distances apart, and the drivers were told to drive normal. There's no stop signs, no stoplights, no reason why a traffic jam should emerge. But if I start this video, what'll happen, they've slowed it down here, is you see a traffic jam forming and propagating backward relative to the direction that the cars are moving. And so I think that this is so cool. And whenever I am driving in traffic, or especially when I'm boarding an airplane, I think about if I were to modulate my speed differently, how would that disrupt or lead to traffic jam forming? And so there have been folks at Temple University who have actually added to this experiment. They've put driverless autonomous cars on the road, and they found that if you add one single autonomous vehicle and modulate its speed, you can stop the traffic jam from forming and save everybody gas money. So that's kind of cool. Another example of a complex system that we might be more familiar with is flocking birds, right? Here you see these birds, by interacting with one another, they're tracing out this kind of collective group dynamic. Another example of a complex system would be instead to look at disease transmission. So I think that this is one that we were all perhaps unfortunately more comfortable with than we want to be. So you remember these kinds of ideas about flattening a curve. So you can think of a disease as being a complex system in the sense that it's propagating between people and how all of our interactions are leading to this emergence of a disease dynamic. Now, these are all examples at the scale of organisms, but you can also have complex systems at much smaller scale, and specifically at the cell scale. So I'm going to start this video here. And this is a video of a pattern forming on the skin of a fish. You can see time ticking up. This is a growing fish skin. And if you look closely, you can see these black points moving around and organizing into the pattern. And so really those cells, just like people in a room, are moving around and creating stripes. Again, I think that this is pretty darn remarkable. So across all these different applications, whether you're looking at drivers and cars, or birds in a flock, or people, for example, in a crowded room or in a country, or cells in the skin of fish. And here you see specifically a black cell leading the way and a gold cell following it from that fish skin. From a mathematical perspective, they have similar features that we want to describe. So for example, there can be things like, in all these cases, there are multiple types of agents. Usually there's a continuum of different types of agents, but we might want to simplify it from a mathematical perspective, at least. In the car setting, maybe there's cautious drivers and more frustrated, angry drivers. Maybe a driver's opinion and emotions can change in time. In a disease setting, right, we heard about different models in the news. The simplest models would have folks who have a disease or who are susceptible to a disease, but you can have much more different types of dynamics. And then in the skin of this fish, right, you could say, at least here, we can certainly see black cells. There are also gold cells present in the skin. So across the board, in all these settings, I think the broad goal from a math biology perspective is that we want to understand how the agents, how the individuals are interacting normally. And then we want to understand how they are interacting to form altered group dynamics. And that might have applications in the disease setting related to control and dissipating a pandemic or dissipating a traffic jam. And in the developmental setting, it might have applications related to understanding mutations and understanding the emergence of disease in organisms. Okay. So in terms of today's talk, I'm going to talk about three separate things. The first is going to be what I like to think of as using mathematics to comment on biology. So this is one way of doing mathematical biology, and it's specifically going to be related to modeling fish patterns. The second part is going to be what I'd say using biology to inspire new types of mathematics or new mathematical questions. Sometimes when you sit on a biological application for a long while and you're thinking mathematically, it can help you think, you're thinking from a different perspective. And so it can give rise to interesting mathematical questions. And so that will specifically be related to quantifying fish patterns and something called topological data analysis. And then the last part of my talk, where I'll spend the least time, is to say, okay, let's move beyond fish patterns and look just very briefly at disease transmission and then how it relates to some work that I've done with students on opinion dynamics. So I've highlighted my collaborators on these parts of the talk below. And everybody who's italicized is an undergraduate student. All the names are listed from left to right. So really you can see that this is a large number of undergraduate students, predominantly undergraduate students have been involved in this. And several of them have been freshmen. Some of them were first-year freshmen in these projects. All right. So let's jump into it. The first thing I want to talk about is modeling, and specifically modeling patterns in fish skin. So this is that fish that you saw before. This is what it usually looks like. It's called zebrafish or Danio vireo. And if you take a closer look at its stripes, you can see that there are those black cells There are also other types of cells present in the fish skin. They are these silver or blue iridescent iridophores. They're harder to see. You might see these kind of shiny flecks in the skin. There are also orange or yellow xanthophores. Again, they're harder to see than the black cells, but you can certainly see a large kind of region of orange and gold right here. These are the same types of pigment cells that are present across fish and reptiles. And so what you see in this video here is a timeline of normal wild type development. It's unfortunately grayscale, but this is the pattern forming as the fish is developing over the course of several months. And so the reason folks study this is that they want to understand how do all of these different brightly colored pigment cells organize to produce a pattern and create these nice stripes. But the other reason that mathematicians and biologists are really interested in this has to do with mutants. So these are all different zebrafish that don't have stripes. They're all mutant zebrafish, and they're all associated with a specific mutation and a specific gene. And really what's cool about zebrafish is we are actually ourselves, you could say we're 70% zebrafish genetically, and 85% of human genes that are associated with genetic disease have a counterpart in zebrafish. So this is a place where the biologists know that a mutation in, say, the leopard gene creates a spotted zebrafish. They don't know how that change in gene translates into a different way in which the cells are behaving. Are they moving slower or faster? Or are they dividing more? What's going on? And really, that's where a mathematical modeling perspective can step in. And that's kind of the pie in the sky, decades down the line goal that drives all this work. We want to say, in this fish that is closely related to humans, can we understand how differences in genes are related to differences in cell behavior, are related to differences in phenotype or visible animal characteristics in the skin of the fish? And so if you want to do this from a modeling perspective, there's lots of different ways to do that. And really, that's the first question that one hits when building a model is what type of approach to use. I'm just highlighting three. I wanted to introduce these. The first is a continuum model. For those of you who've seen partial differential equations, this is a model where you don't see the individual cells anymore. Instead, you kind of zoom out, you go further away from that picture. Instead, what you'd see is some continuous density of gold and some continuous density of black. And you want to understand how are those changing in time. And so one way of doing this is to build what's called reaction diffusion equations. And really, this started with someone named Alan Turing. If you've seen The Imitation Game, then you've heard of him. If you have not heard of Alan Turing, he's someone to Wikipedia and look up. He was really an incredible person. But he had this kind of amazing idea about pattern formation that led to some of the first papers in zebrafish that I read when I started this project. And so his idea was that two substances that are interacting with one another and diffusing, that means spreading, just like heat spreads in a room, can create a pattern from basically nothing. And so the idea would be, if you think of this as being space, and this as being the density of two types of cells, say black and gold, his idea was if you start with just a uniform amount of one substance, the black substance, and just a little blip of gold, just a little tiny imperfection, then what will happen is if you assume that gold part is what's called an activator, the activator activates itself. It also activates the black curve in this region, which is the inhibitor. Then what's going to happen is you get a pop-up of more gold in that region and also more black. And then the assumption was that that black density needs to spread out more quickly than the gold. So if it spreads out more quickly, that the next time step, you're going to have something that looks like this, where you can see the black has spread out. And then the key is that that black density inhibits the activator. And so basically you end up staying right at this point. This high peak of activator is able to keep itself activated, but at the later places and position here, there's a high enough density of that black, that inhibitor curve, that no more activator is present. And so this kind of creates a pattern that remains there. This is an example of a system of reaction diffusion equations that can create this type of dynamic. I'm not going to go further into it. I just want to highlight that these are the types of things that folks study when they're interested in pattern formation using partial differential equations. And these are things that folks can prove theorems about and do things like analysis and bifurcation analysis of, if you're interested in that. Now, two other types of approaches, which are more computational, but also take kind of a closer scale look at the cells themselves, are what I'll call lattice-based or cellular automaton models and agent-based models. So in this lattice-based setting, what you're doing is saying, I want to understand what the individual cells are doing. So you'd say, I want to break my domain up into a checkerboard, and each square in that checkerboard can be black. If it has a black cell in it, it can be gold if it has a gold cell in it, or it can be white if it's empty. And so one thing that we know about the way that these cells interact in the skin is that black cells move away, they lead the way, and gold cells follow. Let's see. Okay, great. Yeah. Thanks, Lee, for sharing that about Alan Turing. Great. And gold cells follow. So in one way that we can do that from a computational perspective is to say, okay, if black cells want to move away from gold, then if you're looking at a pattern here, you look at this black cell, it has 3 gold cells here, it has an empty space there. Then in a lattice-based model, what you'd say is that the next time step, there's some high chance that that black cell will pop over into the empty space. So it's kind of a stochastic rule for cell movement. If you wanted to count for, say, cell death, one of the things that we know biologically is that cells die off when they are surrounded by cells of the opposite type. So again, if you have some black cell here and you look at its 4 nearest neighbors and they're all gold, then at the next time step, you might kill off and remove that black cell from the domain. So this is one type of modeling approach. The last approach I'll mention is this agent-based approach. And I think that the best way to describe an agent-based model is with a video. So I'm going to start this video in just a moment. This red scale bar is 500 micrometers. And this rectangle is going to be growing in time because the fish is growing itself. And each point here is a pigment cell. And so if I start this video, you can see the number of cells is changing and growing. The domain is growing and the cells are moving around. So in an agent-based setting, what you're doing is prescribing a differential equation for every single cell that describes its motion. If you've taken kind of an ordinary differential equations class, then this would be something that you could work with, or even a physics class. Really what we're saying is that the velocity of the ith black cell is equal to some function. You can think of this as being force equals mass times acceleration, only we're in a simplified setting where velocity and force can be kind of related. So you have differential equations for cell movement, and then to account for, say, for example, cell death, it's kind of the continuous space equivalent of that lattice space setting, where here, if you have a black cell here, and you want to say that black cell is surrounded by orange cells, it'll probably die off. You just build a ball around the black cell, count up the number of gold cells in the ball. If it's too high at the next time step, you remove that black cell. So this is a kind of general structure. Now, the benefit of these types of models is that they really allow you to work on the same time and length scales as experimental data. They really look like the development of the fish. And you can see this number counting up here. This is in days post-fertilization. So this is how old the fish is. It's, in this case, about a month old. And really, this is the approach that we're going to use because it allows us to work so closely with the biology. Now, in this video, you might notice that there are lots of different colors. There are not just black and gold. That's because we were kind of simplified it in these pictures. My very first models of zebrafish were models that had black and gold cells only. As you build kind of more and more years working on an area, you start to build more complexity and to account for more realism. So I'm going to talk just a little bit about these types of models. So this is that model summarized on one slide. So this is an agent-based approach. That means we're considering these individual cells, just like your XY coordinates to room, moving around in 2D. We'd say m sub I would be the center of the ith black cell, just as XY coordinates. X sub d sub I would be the center of the ith orange cell, and so on. When the fish is young, it might have 100 cells. We simulate about 2 months of development, and by that point, it has about 10,000 cells at the end time. It takes about 3 minutes on a laptop to simulate 2 months of development. And then the cells interact in really 4 main ways. There's cell movement, so we talked about that before. You see this black cell leading the way and the gold cell following it. We're going to describe that using a couple differential equations. And then all the other interactions in the fish skin, cell birth, so changes in number of cells through cell division or differentiation from precursors like stem cells, cell death through competition, and then another type of interaction, these changes in cell type. Really, you can think of this as a cell kind of changing its color and changing how it chooses to behave. Those are all going to be through stochastic discrete time rules. And so I want to mention here that when you're building a model, it's important to understand from the very beginning, what is the question that you want to answer with the model. In the case of this model, I'm not going into too many details, but really the key here was that when we built this model, cell movement and cell birth and death were very well understood biologically. And so this was a place where we were kind of taking known biology and plopping it into a mathematical framework. Where we were really commenting on current unknowns was in this case. The experimentalists thought they could see the cells changing their color in the skin, and they had no idea what was causing that. And so really that's what we wanted to shed light on. So I'm just going to give you a flavor of these different rules. This is one of the differential equations for the velocity of the ith black cell, right? So I said m sub I is the position of the ith black cell. So dm I dt is the velocity, right? The derivative of the position. And this just, it looks complicated, but all this is saying is sum all the forces that black cell feels from all the other black cells. It depends on the distance between the black cells, the sum of the forces it feels from gold cells, and the sum of the forces it feels from silver cells. Specifying parameters and models is always difficult. In this case, we had some measurements of things like cell-to-cell distances and cell speed that we could use to determine what our parameters should be. But this is kind of the general form, and you have one differential equation per every single cell. I use MATLAB to simulate these. If you've seen Ford-Euler, if you've done a differential equations class and seen Ford-Euler, that is the method that we use to simulate them. Okay, and then for all the other cell interactions, this takes the form of the stochastic discrete time rules that I told you about before. Just to give you an example of one of these, this was an experiment that was done by Shigeru Kondo's lab, where what they did was in a living fish, they took the fish and used a laser to remove the gold cells sandwiching a black stripe. And then they watched what happened several days later. It's a pretty cool experiment. And what they found was that several days later, a large portion of the black cells died off. If you do the same experiment in reverse, where you say, kill off the black stripe sandwiching a gold stripe, what happens to the gold cells? The gold cells don't care. And so the conclusion was, somehow or another, those black cells are relying on a survival signal that's coming from long range in the gold stripe regions. Now, it hasn't been linked biologically, but one possibility is these types of long legs. So this green blob is a black cell, and it's reaching out a long leg into the gold stripe region, up to 5 cells away, quite long. And you can think of that as being a cell that's reaching out a leg, trying to find a gold cell so that it can pass a signal. It doesn't find a gold cell, and maybe that means it dies off, right? So that kind of dynamic we take into account in the model by saying at every single time point, you look at every single black cell in the domain, and you construct a red annulus around it that's kind of modeling what cells could that cell interact with through its long legs. And you're saying count up the number of black cells in that annulus. If it's larger than the number of orange cells in that red donut, then with some probability you kill off and remove the black cell in the center. So this is kind of the general structure of all the rules involved in cell birth, cell death, and cell shape changes. You're either evaluating a precursor for possible birth or differentiation of a new cell at the center, you're evaluating an existing cell for cell death, or a cell for changing its color. And it's all going to depend on inequalities in the types and proportions of cells in either its local neighborhood, that blue disk, or that red annulus donut region. So once you have these types of rules, I'll just show you what it looks like when you simulate it. This is going to be our wild type pattern formation. This is what the zebrafish normally looks like. Again, this is going to be how old the fish is in days. So right now it's 21 days old. And you can see the pattern forming in the skin. This is 2 months of development simulated in about 3 minutes. This is the Pfeffer mutant. That's the German word for pepper. It has no gold or yellow cells. And you can see that it creates this pattern that's kind of blue spots, roughly aligned in stripes, and a silver background. And then this last mutant here is nacre. And before I get to the kind of second component of my talk, one thing I want to mention here is if you're looking at all these patterns and saying, is this one a good match for that one? It looks kind of different. This one, maybe you buy that this one looks like this fish. Or if you're looking at this one and saying, well, these look very, very straight. Those stripes look pretty straight. These are kind of curvy. I think those are exactly the right questions to be asking. And so this really highlights how, when you're kind of going from a biological perspective, how you can raise new mathematical questions. And really the questions here are, how do we take messy patterns and develop methods for quantitatively describing the relationship between messy patterns so that we don't have to rely on kind of an eyeball norm subjective decision about whether these mutants are good matches for the biological data. Now, you might be saying fish aren't rectangles. And so I wanted to highlight some work that was done by undergraduate students. So these were 2 teams of undergraduate students across 2 summers. One team looked at pattern formation on fish fins. They were the first group to ever do that. And then the other said, okay, building on that, let's go ahead and look at the full body. So here you see 2 simulations where we've got pattern formation on the body and on 2 fins of the fish. And when we first built these domains, we did it with no head because the eye here kind of messes up the patterning process. I thought it was so uncute without the head. So we added back the head in post-processing here. Okay, so you can see one of their papers here, the other is in progress. Okay, so the other, but I'll just highlight this as well. This is another example of modeling work in a slightly different setting with undergraduate students. You might also say zebrafish patterns, you used all those brightly colored symbols to mark the cells. That's not what it looks like in the real fish. So Caroline and Annapurna have been working to actually develop new methods for plotting our simulations. So this is we like to think of it as our hashtag fake fish. This looks real, but it is actually one of our simulations that has been presented in a realistic way. And this is really, I think, a combination of math and art and computer science. The other example I wanted to highlight here, if any of you have siblings or children who you like to talk about math with, this was work with a student, Blake Sherman, who had never had any programming experience. And what he did is develop a website. And together we built this craft to highlight to children an example of how to explain kind of pattern formation in mathematical biology through zebrafish. So like I said, I'll put these slides on my website down the line with all these QR codes. But if you have children yourself or siblings who you want to share this with, this simplifies things. All right, so with that, I'll go into the next kind of major theme of my talk. That first part was where I intended to spend the most time. So really, all these different patterns kind of raise this mathematical question of, I showed you these simulations, and depending on how we're all feeling, it's a Sunday, some of you have been in Zoom for a long time. I think if I asked the whole room whether or not you think this simulation is a good match for this fish, if we took a poll, some would agree yes, some would agree no. Maybe if I asked you tomorrow, your opinion would change, right? And the whole critical issue here is that it is a subjective call. And this is not just an issue because it introduces that kind of subjectivity. It's an issue because it limits our kind of the power that our models can provide to the biology. Because if understanding what parameters lead to different mutants relies on an expert viewing the pattern and making a qualitative call, that's a big bottleneck in the process. So really, we want automated methods that allow us to measure the difference between these two images and to quantitatively describe them. And this is an issue that is much broader than fish patterns. So in this row here, I'm showing you in red all sorts of different patterns in biology, real patterns. And in blue, I'm showing you different models that are meant to be a good representation of those images. This ranges from cells to mussels on a beach to trees in vegetation patterns. And really across the board, you see kind of similar patterns, but really we want to be able to move past this kind of eyeball norm. I'm showing you only one single time point in the patterning process. What about the other times, right? I'm also showing you only one single example of each of the real images and only one single example of the simulation. A lot of these simulations are stochastic, right? So we want to not just look at comparing one and one, we want to compare thousands and understand, are we building models that capture realistic variability? And so the method that we're going to use here is something called topological data analysis, specifically persistent homology. And so I'll say here, I'm actually, like I said, my PhD was in applied math. So I actually never took topology and geometry classes. So some of y'all will have more experience with that than me. But really, when you're sitting on a biological application and you're really interested in the biology, it drives you to go in all these different mathematical directions. So I just want to introduce a little bit about this method, persistent homology, that we're going to use. And so broadly, this is a means of measuring the shape of data. And so your data can be in any form. It could be the position of pigment cells in fish skin. It could be the position of people in a room. It could be all sorts of things. It doesn't have to be two-dimensional. So in this case, suppose our data is these black points here. One way of computing persistent homology is to put a ball around each point and then slowly increase the size of the ball. And then the key is that there's nice computational resources out there that will tell you, as a function of the ball size, what is the number of connected components, that's clusters, and what is the number of holes or loops in your data. And so at this case, you see the number of connected components is just the same as the number of points. At this point here, we see there are clearly 4 clusters, which kind of makes sense, and one tiny loop that's going to disappear as soon as we make those balls a little bit bigger. And then finally, at this point, we have one single connected component and one hole or loop in the data. And so really, we want to take this idea of measuring connected components and loops and use it as a means of measuring fish patterns. And this is a very active area in mathematical biology. I'm highlighting a few other references where folks have used these methods to look at other types of biological data. So that's all fine. But the issue is, if I give you some data and say, okay, how many connected components or how many loops are in that data? Tell me using persistent homology. You'd say, well, give me a ball radius. And then I can tell you how many holes or loops are in the data. And so really the key is that we're going to say, okay, the number of loops that are going to be important to us are going to be the loops that persist for a large range of ball sizes. And so to give you a sense of this, this data looks like a figure 8. So we might all say, what is the shape of that data? It should be one connected component and 2 holes, because that's what a figure 8 looks like. If you were to put balls around that data and you zoom into this yellow square, there's a tiny little hole there, but that hole is going to close up quite quickly, right? But at some point between R1 and R2, we see the emergence of one single connected component and 2 loops. And those 2 loops are going to remain all the way up until right before R4. So the way that folks look at this sometimes is using what's called a barcode. The y-axis here doesn't really mean anything. What's important is this x-axis. And what you do is start a bar at the ball size that the loop first appears in your data, and then end the bar where it disappears. And so you'll notice there's all these tiny bars that has to do with these small loops. And some point between R1 and R2, we get 2 large bars that emerge and remain all the way up until right before R4. That's these 2 holes here that are meaningful to us. And so what we're going to say is how many loops are present. It's the number of loops that remain for a long enough range of values. And now that's a question of what is long enough. That's something some additional undergraduate students have been looking at actually, and I'll highlight that a little bit later. All right, but let's go ahead and take this and apply it to fish patterns. In our case, our fish is actually not a rectangle, it's a cylinder. The domain is periodic in X. And so if you want to apply persistent homology to the positions of, say, your gold cells, in this case, then really you'll notice that in that case, the presence of a stripe in your image is really the presence of a loop on that cylinder. And the presence of a spot in an image is the presence of a connected component. So directly from persistent homology, we get our hands on the number of stripes and the number of spots in an image. If you want to get your hands on, say, stripe width, you'll notice that the point at which these balls intersect and those two connected components merge, that ball radius is half the width of the intervening stripe, right? So that gives you information about stripe width. Now, once you know how many stripes are in your image, you can do things like cluster your cells into that number of stripes. That's what we've done here. And then you can trace out the boundary of the stripe and measure the length of that red curve relative to the length of the domain. And that gives you information about stripe curviness. So let's just take these types of tools and apply them to simulations of our fish patterns. So this is simulations. This is 1,000 simulations of wild type and 1,000 simulations of these 3 different mutants. And so what I'm showing you for the first time is distributions of gold stripe width or distributions of stripe curviness. And before this, we were just by hand looking at all these things, like I'm doing the eyeball norm, or we can quantify, say, the difference between these different mutant patterns that have only been defined qualitatively before. So for example, this knacker pattern, we say that its spot size is more variable. It's also larger than these other 2 mutants. So you can think about comparing these to distributions from the biological data itself. And that's something that we're working on as well. But this really provides a larger scale perspective on the models. Now I'll highlight 2 directions that undergraduate researchers are going in and will continue to go in this kind of quantifying patterns theme. I mentioned that there are different types of models for pattern formation in zebrafish and other settings. 2 of those approaches that I talked about earlier were these agent-based models, and then the other being these cellular automaton models. Electa and Angela have been adapting those persistent homology methods to understand the relationship between different modeling approaches, because it's important not just to know how models relate to biology, but also how models relate to one another. And so you can see, for example, that this cellular automaton lattice model has a narrower stripe width on average, but also a lot more variability than the agent-based model in blue. This is across 1,000 simulations. We're saying what fraction of different patterns have different stripe widths. So their manuscript, I'm realizing I don't have the reference on this page, but I'm happy to share it if you'd like. It's coming out in SciM Dynamical Systems soon. Now, the last thing I want to talk about in quantifying patterns is just to mention that I'm really excited to be part of the SciM's undergraduate research program in 2024. So it hasn't been announced yet who the mentors are, but I will be one of the mentors. So if you watch this space, the applications are going to open up in, I think, around December 2023. This is also a QR code to that link, but this is an REU open to students for summer 2024. We're looking for folks who have hopefully some coding experience, but you don't have to have experience with differential equations or linear algebra or calculus. And really those projects are going to be related to quantifying patterns either from real fish or from plant biology or simulations. And we're interested in using some of these topological data analysis tools, but also something called optimal transport to look at patterns like these. So these are patterns that are from real fish. There have been some image processing where really the fish is black and gold, but I've binarized it and made it black and white. And we want to understand, can we compare these patterns, many different types of patterns like these, and understand how closely related or different they are. So keep an eye out on that program if you're interested in some of this stuff. I'll mention SciM Simons is also they're going to have other types of projects as well. It won't just be this one. So it should be a great thing to look out for in applied math. All right. So the last thing I want to do, just for completeness, is highlight one other type of direction that we've gone in with undergraduates. And this is work on a much higher scale. So we're going to kind of drop cells and move to the scale of individuals. And the individuals we're going to look at in this case are people interacting, in particular on college campuses, and understanding disease spread in that setting. So this work with students was based on trying to understand the dynamics of the flu on college campuses. It happened way before COVID. We looked at this in 2016. So the general modeling approach that we're going to use in this setting is what's called susceptible, infected, susceptible models. These are models that are useful for diseases that you can get again multiple times. And the general idea here is that you have two groups of people in the simplest form. You have a group of people that is susceptible to some disease and a group of people that is currently infected with the disease. And then you can, if you are susceptible, become infected by interacting with an infected individual. And then the other assumption here is that infected individuals recover and go back to being susceptible, and then they can catch the disease again. Hopefully not, but that can happen. All right, so there's different ways of looking at epidemic modeling, just like there's different ways of modeling pattern formation. And a lot of times it depends on what your goals are and what type of data you have available. I just wanted to highlight these two examples. In the first case, you're looking at individual people. So this is a network or graph. These are nodes and the lines are edges. So you'd think of people as being each point here, and the edges representing that they've had an interaction with the person that they're connected to. And you see I've colored these either purple if they're susceptible to a disease, or blue if they're infected. And so if we start with one single infected individual in the network setting, you'd say that that infected individual has some chance at the next time step of passing the disease on to the people it interacts with. So this type of modeling approach would use things like linear algebra and network science, network analysis. The other type of approach that I want to highlight here and that we'll use for the next couple of slides is, again, a differential equation approach, specifically an ordinary differential equation approach. And in that case, you say, zoom out. I don't know or need to know what any specific person's infected or susceptible status is. I just want to know how many total people in the population are susceptible or infected. So in that case, if you want to write down a system of differential equations that describes the number of susceptible folks in the population and the number of infected folks in the population, if you assume it's kind of a closed population, there's no changes in the number of people present, then in this case, you say that the kind of transmission occurs when a susceptible person interacts with an infected individual. So SI here, the brackets mean that's just the number of interactions between susceptible infected individuals, and beta has to do with kind of the rate of transmission. And then you can have infected individuals that go back to being susceptible, and that depends on some parameter gamma, that's your recovery parameter. So if you just take this system of kind of this image and translate it into a mathematical model, you get something that looks like this. You might notice again that term with the brackets, SI. What is that? Well, usually when folks are building the simplest types of models, they simplify that and assume that there's kind of uniform mixing, that everyone is equally likely to interact with everyone else. That's usually not true. But if you take that as an assumption, then you end up with this system of differential equations. So this is telling you what is the kind of evolution of susceptible individuals of time and the evolution in infected individuals in time. And so I wanted to highlight this work by these two undergraduate students, Anne and Ruby, who worked on this type of project a good while ago in 2016. And really they were interested in working with disease transmission in a college campus. And so they wanted to look at disease transmission in networks and really account for the different kind of places where people interact. And so they wanted to build networks that could represent say, your home or your dorm, and a network representing, say, your interactions in a classroom setting. So the idea would be, in this case, they thought in a dorm, they needed to model this. So they assumed maybe folks have a couple of roommates. So it's everyone's connected to everyone, and it's just small networks that are disconnected based on this being one dorm room, this being the next dorm room, maybe this being a family, and so on. And then for classes, they thought this would be larger, maybe in a relatively small class, there's only 15 people. And again, they had to develop a means for modeling this. In this case, they assumed that everyone was likely to interact with the instructor. And so that, again, developed a certain type of network. And so then they simulated diseases on these different networks, and we're looking at the size of the disease, the epidemic, as a function of time when you take out different types of networks. So suppose you say, cut out your social interactions or cut out your interactions at school. How does that impact the spread of the disease? And you can see that it certainly changes how large of a peak we get. So the last thing I wanted to mention here is just some other work with students that goes more towards a social science side. So this is just to say that, I think there's a fair number of folks who work in mathematical biology who also do a little bit in social systems. And so I wanted to just highlight here that you can take these same types of models. What these models are at their core are what's called compartmental models. They're just saying that you can separate people into kind of simplified groups and then understand how people change what group they belong to. And so you can think about that in a political system as well, where you could have folks who maybe belong to an undecided group for a political party, or belong to a Democratic group in a political party, or belong to a Republican group in a political party, or you could have more groups. We're just going to simplify in this case. But I'll just highlight here that Ryan, Monkey, and Manas, again, 3 undergraduate students who were first-time undergraduates when they started working on this project, actually took this kind of general modeling approach and used it to produce forecasts of the 2022 midterm elections. And so I've highlighted their website here. I've also put a link to their website. If you go to that website, you can see all of their code. We've made it very much publicly available if you'd like to try out forecasting elections yourselves using some of these models. Now, in the interest of time, I'm going to skip all this. But again, this will be on my website once I share these slides. These are different resources for getting started in modeling, or a contest for modeling if you're interested in that you can do during the semester. These are some resources on how to find mentors in mathematical biology at the undergraduate stage or the graduate stage, and then just some resources for getting started in topological data analysis, or specifically in pattern formation in zebrafish, or hearing about other areas in mathematical biology. So, for example, the Society for Mathematical Biology's YouTube channel is a great resource that has different lectures. All right, so with that, I want to acknowledge my collaborators again. I'll be in one of those RAU networking rooms later on, and I'm happy to take questions or comments. Thank you all.